Okay, let's dive in. We're looking at some source material today that's all about taking a basic simulation of the cochlea, that amazing spiral thing in your inner ear,
Hmm.
and making it much more, well, biologically real.
Right. The mission really is to pull out what these sources say adds that crucial biological accuracy.
Yeah.
And honestly, some of the details about how we actually hear are pretty surprising.
Exactly. And what's interesting is these sources aren't just describing the ear, they're actively critiquing a simpler simulation and saying, look, here are three specific ways to make this thing way better, way more like the real deal.
Yeah, it's like bridging the gap, isn't it?
Yeah.
Between a sort of cartoon version and the actual biological engineering masterpiece in there. The sources basically highlight what gets lost in simplification.
Totally. So the first big area the sources jump on where that basic sim falls short is how it maps frequencies. Like a simple model might just have one slider, and changing it makes the whole simulated membrane wave differently, but uniformly.
Yeah, exactly, like one size fits all.
Yeah.
But the real basilar membrane inside your cochlea, it's way more sophisticated. Think of it like a ribbon that's not the same all the way along.
Okay.
It's narrow and pretty stiff right where sound enters, the base. And then it gradually gets wider and much more flexible as you go towards the other end, the apex.
Wow. Okay. So it's not just passively vibrating. That physical difference means high-pitched sounds make the stiff bass vibrate the most.
Precisely.
And the low pitches, they travel further down and hit the sweet spot on the wider, flexible end.
You got it. That's the tonotopic organization they talk about. Different pitches physically peak at different places. So the sources say, okay, fix the simulation. You need a function where the frequency input determines a peak vibration spot.
Right.
High frequencies peak on the left, low frequencies on the right, maybe, and then the amplitude for each little simulated hair cell depends on how close it is to that peak.
Ah, so only the cells in the right place for that pitch really get activated strongly.
Exactly.
And why this matters for you listening is that this is how your ear sorts sound by pitch before it even becomes a nerve signal. It's mechanical pre-processing. Super clever.
It really is. Okay, so that's the physical sorting. The next level of complexity the sources tackle is the cells themselves. A basic simulation might just have, one type of generic sensor cell.
Right, doing everything.
But the real ear, it's got this amazing division of labor. You've got the inner hair cells, just one row of them. Those are your main sensors. They're the ones actually sending the signal to the brain. Think of them as the primary microphones.
Okay. The main players.
But then behind them, you've got three rows of outer hair cells. And these, the sources explain, are like the ear's built-in amplifiers, especially for faint sounds.
Amplifiers. How?
They don't just sit there, they actively move. They physically push and pull to boost the vibrations locally, right where they happen.
Wait, they physically move to make quiet sounds louder? That's incredible. So that's the cochlear amplifier people talk about.
That's it. It's this biological motor giving those tiny vibrations a little nudge so the inner hair cells can actually detect them.
Wow.
So the sources suggest adding these OHCs to the simulation, 3 rows. And when the simulated sound is quiet, you'd see these little OHCs maybe visually twitch and actually add a bit to the vibration value for the inner hair cells nearby.
That is so cool. And that amplification explains how you can hear really faint stuff, right? It expands the whole range.
Absolutely. It's active biological gain control. Amazing stuff.
Okay, so we've got physical sorting by pitch, and we've got cellular amplification. What's the third big thing the sources critique in basic models?
It's how the output is shown the neural signal. A simple sim might just have a light bulb that flicks on when sound is detected. Sound? Yes, no.
Uh-huh. Very basic.
But your brain needs more than that, right?
Yeah.
It needs to know how loud the sound is. And the way the auditory nerve encodes loudness isn't by sending a bigger electrical zap.
How then?
It's the rate of firing, how many nerve spikes per second. A louder sound makes the nerve fire more often, more rapidly.
Ah, right. Like tapping faster means more important. Okay.
Exactly. So the sources say, ditch the light bulb. Replace it with, say, a little scrolling graph showing these nerve spikes. And you use a variable from the simulation, maybe one that tracks the total number of activated cells, let's call it total activation, as a proxy for loudness.
Gotcha.
Low total activation means a quiet sound, so you draw spikes slowly, far apart on the graph. High total activation, loud sound, means you draw spikes really quickly, bunched together.
So the simulation would visually show how intensity information is actually packaged for the brain, not just on-off, but a rate code.
Precisely. You're visualizing the neural encoding of loudness.
Okay, that makes a lot of sense. So pulling it together, the sources give us these three key upgrades. Mimic the physical pitch, sorting the tonotopic map, add the two types of hair cells, especially those outer hair cells during the amplification dance, and then model the neural signal as a firing rate, not just an on-off switch.
Yeah, those are the big three. And implementing them takes the simulation from a simple concept to something that starts to reflect the, frankly, incredible biological engineering happening inside your head.
It really drives home how much complexity is packed in there. It's not just a passive receiver at all.
Not even close. It's active, it's dynamic, it's intricate.
Which leaves us with a thought for you, the listener. If hearing something so constant has these layers of, well, almost hidden complexity revealed when we look past a simple model, what other everyday biological things, systems that seem simple on the surface, might have equally surprising mechanisms at work? Something to think about.